Fertilization in humans is internal. It takes place inside the body of the female. Semen, which contains the sperms, is introduced into the female genital tract during the act of copulation. The deposition of sperms in the vagina is called insemination. Human sperms are motile and rapidly swim towards the ovum, lying in the fallopian tube. The sperms pass the cervix, enter the uterus, and finally reach the ampullary isthmic junction of the fallopian tube. This is the site where the ovum lies and fertilization takes place. When a sperm meets the ovum is in the secondary oocyte stage. It has not yet completed its second meiotic division. The fusion of sperm and ovum is called fertilization. For fertilization to take place, the sperms must be released in the vagina during the period that the egg is alive, that is, within 24 hours after ovulation. The sperm has to pass through the corona radiata and the zona pellucida to reach the plasma membrane of the ovum. When it reaches the zona pellucida, the acrosome releases its contents of lytic enzymes. These enzymes dissolve the zona pellucida. The sperm plasma membrane then fuses with the egg plasma membrane. This fusion triggers the secondary oocyte to complete its second meiotic division. The second meiotic division is also unequal and leads to the formation of a second polar body and a haploid ovum. The nucleus of the ovum is now called the female pronucleus. The cortical granules present below the egg plasma membrane release enzymes which harden the zona pellucida and block further entry of sperms. The sperm nucleus now enters the cytoplasm of the ovum. The sperm nucleus is now termed the male pronucleus. The male and female pronuclei come together and fuse. The male and female chromosomes mix. This event is called karyogamy or amphimixis. Fertilization is thus completed. The fertilized ovum is called the zygote. Identical twins are formed when the zygote splits into two. Each split portion then develops into a complete new individual, and both are exactly identical to each other. On the other hand, non-identical or fraternal twins are formed when two separate eggs develop in the mother's body, and they get fertilized by two separate sperms. The zygote is diploid since it contains two sets of chromosomes. Human beings have 46 chromosomes, out of which 44 are autosomes, and the remaining 2 are sex chromosomes. The human male contains 44 autosomes plus 1 X and 1 Y chromosome. So, 50% of the gametes formed by the male will have X and 50% will have Y chromosomes. The human female contains 44 autosomes plus 2 X chromosomes. So, all gametes formed by the female will have X chromosomes. The sex of the baby depends on the sperm that fertilizes the ovum. The ovum contains 1 X chromosome and 22 autosomes. If the sperm that fertilizes the ovum carries an X chromosome, then the zygote will have 2 X chromosomes and the embryo will develop into a female offspring. But if the sperm that fertilizes the ovum carries a Y chromosome, Then the zygote will have one X and one Y chromosome, and the embryo will develop into a male offspring. After fertilization, the zygote moves down towards the uterus, all the while undergoing a series of mitotic divisions. This series of cell divisions, by which a single fertilized egg cell is transformed into a multicellular blastula, is called cleavage. The cells formed by cleavage are called the blastomere. Cleavage transforms the single-celled zygote into the many-celled blastula, then the 16-celled morula, and finally the hollow blastocyst. The blastocyst contains an outer layer of blastomeres called the trophoblast layer and an inner mass of blastomeres attached to the trophoblast. The inner blastomeres together constitute the inner cell mass. The trophoblast layer attaches to the endometrium, and the cells of the inner cell mass continue differentiation and development. The blastocyst burrows into the uterine lining. This occurs about 7 days after fertilization. The embedding of the blastocyst into the wall of the uterus is called implantation. The blastocyst sinks into a pit formed in the endometrium. The endometrium grows around it and the blastocyst gets completely buried in it. After successful implantation, the embryo continues its development.
Hello everyone, Jai Hind student once again. Today I am here in front of you with a very interesting topic, that is the cleavage and the, you can say, implantation. Listen, in my last video I have explained you the process of fertilization. You must be remembering. Clear? Then what is the result of fertilization? The result of fertilization is the formation of zygote. Clear, student? Now what is the fate of zygote? What happens to the zygote? Clear? So now let us discuss. Clear? The fate of zygote. Yes, the formation of embryo and then the fetus. Clear? One thing, student, I want to clarify here that now there will not be any meiosis cell division. Clear? So whatever the cell division will take place, it involves the mitosis cell division. Clear? This is a general question which is generally asked in the NEET examination. Clear? So now just after 30 hours, clear, of fertilization, clear, for 30 hours the zygote remains as such. Some physiological changes takes place in zygote, and now after 30 hours the zygote starts dividing. Clear? It divides and redivides and form a multicellular ball called as the blastula. Clear, student? Here you can see very easily, you can identify all the stages. Clear? This is the zygote. The ploidy of the zygote is 2n. Clear? Now the zygote undergoes cleavage. Simply means division of zygote. Clear? So zygote undergoes the longitudinal division. The first division is longitudinal. The result is the formation of 2-celled embryo. Then horizontal division result in the formation of 4-celled embryo. Then again the cells divides. They form the 8-celled embryo. Clear? Student, these cells are called as the blastomeres. What do we say? हम लोग इन सारी cells को बोलते हैं blastomeres. Clear? These are called as blastomeres. और ये अभी देख सकते हो, this is the layer of zona pellucida. This layer persists. अभी यहाँ पे this layer persists. Clear? And what happens? As soon as the division starts, the zygote starts moving towards the uterus. आप देखो diagram में it is given. Zygote, then this is the 2-celled, 4-celled, 8-celled, 16-celled embryo. Listen, now it start moving towards the uterus where implantation takes place. Listen, so now very soon the 8 to 16 cell embryo is formed, which is in the form of solid ball-like structure, and at that stage we use the term morula. Clear? We use the term what? Morula. Clear, student? It's at this stage it is in the form of solid ball-like structure. Clear? So now some changes takes place. Clear? What are the changes? जैसे हम शर्ट की बाजू में हाथ डाल के उल्टा कर देते हैं, इन द सेम वे ऑल द सेल्स दे आर टर्न लाइक दिस, और टर्निंग होती है देन अ कैविटी इज फॉर्म्ड. क्लियर? एंड एट दिस स्टेज व्हेन कैविटी इज फॉर्म्ड, दिस मोरुला इज नाउ टर्म्ड एज द ब्लास्टुला. क्लियर? और सिंपली वी से द ब्लास्टोसिस्ट. पहले आप देखोगे सिंपल अर्ली ब्लास्टोसिस्ट होता है, इसमें चेंजेस होते हैं. नाउ कैविटी इज फॉर्म्ड इनसाइड. Clear? And now this is called as what? Blastocyst. Clear? This is called as blastula or blastocyst. This is the basic difference between the morula and the blastula or the blastocyst. The cells of blastocyst, they are called as the blastomeres. Clear, student? Now here it is very important to know that the cells which are at the margin, they are called as the trophoblast. ये स्ट्रक्चर बहुत इंपॉर्टेंट है. दिस इज द स्ट्रक्चर ऑफ ब्लास्टुला. क्लियर, स्टूडेंट? ब्लास्टुला. क्लियर? 2 मार्क्स क्वेश्चन में दिस इज ऑफन आस्क्ड इन द बोर्ड एग्जामिनेशन. सो द सेल्स व्हिच आर एट द मार्जिन, दे आर कॉल्ड एज द ट्रोफोब्लास्ट. क्लियर, स्टूडेंट? ट्रोफोब्लास्ट. These cells, they get attached to the endometrium wall of the uterus. You will see here endometrial wall से attach हो जाएंगे and they will help in the formation of placenta. Clear? Clear, student? And they will help in absorption of the nutrition or the nutrient. Clear? Now the red cells which you can see here, लाल वाली सेल जो दिख रही है आपको, these are called as the inner cell mass. Clear? Now these cells further divide and differentiate to form the embryo proper. Clear, student? Question पूछा था what is the function of inner cell mass? तो the cells of the inner cell mass, they differentiate and they form the embryo proper. In future they will form the primary germ layer. Clear? Ectoderm, mesoderm, endoderm, which give rise to the various organs of the fetus. Clear, student? So this is about the, you can say, blastula or the blastocyst. Clear? Here is a cavity which is known as the blastocele. Clear? Here you have to focus. Clear? From board point of view you have to focus. Clear? The two names, that is the trophoblast and inner cell mass, you have to remember. Clear? Now what happens student, इस सारे you can say in सभी process में 6 to 7 days का time लग जाता है. After 6 days, clear? Now the blastocyst or the blastula is in the uterus. Clear? अब uterus की wall से वो stick कर जाएगा. देखिए यहाँ पे मैंने बनाया, you can easily see this is the inner lining. Clear? This is the endometrium, endometrial lining, epithelium. Clear? And this is the, you can say, the blastocyst. This one is the blastocyst. Now it gets attached. Clear? अब ये blastocyst, it gets attached to the uterine wall, endometrial wall, inner wall of the uterus. Clear? And now very soon, 7th day, 6th to 7th day, now what happens? It gets embedded. यहाँ पे देखो, it gets embedded in the uterine wall. अब ये धीरे धीरे आप देखोगे ये यूट्रस की वॉल में अंदर धंस जाता है and the covering develops around it. Clear? Now this is called as the implantation, the process by which the blastocyst gets embedded in the endometrial wall for further development. This is called as the implantation. Clear, student? So this process you can say takes almost 6 to 7 days. ये पूरे process में student 6 to 7 days का time लग जाता है. Clear? So this is about the cleavage and the implantation. Clear, student? So in my next video I will explain you about the formation of placenta and the, you can say, the pregnancy and the parturition. Clear? Thank you very much.